Lac operon. The lac operon is a classic example of a genetic response to an environmental change. Glucose is the preferred metabolic substrate in Escherichia coli. However, when glucose is absent and lactose is available, the lac operon is activated to switch the cell to lactose metabolism. The mechanism of this shift works in two ways. 1. When glucose is low, there is an increase in adenylate cyclase activity. This leads to an increase in the generation of cyclic adenosine monophosphate, CAMP, from adenosine triphosphate, ADP. The increased AMP activates catabolite activator protein, CAP. Activated CAP binds to the CAP site and induces transcription, which increases lac operon transcription. 2. When lactose is high, allolactose, which acts as an inducer, binds to the repressor protein. This binding inactivates the repressor and causes it to unbind from the operator's site, allowing transcription to increase. The combined effect depends on the availability of glucose and lactose. When glucose is low and lactose is available, lac genes are strongly expressed because CAP is active and the repressor is inactivated. When glucose is high and lactose is unavailable, lac genes are not expressed because the repressor remains bound to the operator. When glucose is low and lactose is unavailable, lac genes are not expressed because the repressor blocks transcription despite CAP activity. When glucose is high and lactose is available, lac genes are expressed at very low basal levels because CAP is inactive even though the repressor is unbound. Functional organization of a eukaryotic gene. The functional organization of a eukaryotic gene proceeds in steps from DNA to protein. Step 1. DNA, the coding strand, contains regulatory regions such as enhancers, silencers, and the promoter. The promoter include the CAT box and a TATA sequence called the TATA box, which marks the transcription start site. The gene itself contains alternating exons and introns, with intron-exon boundaries marked by sequences such as GT at the 5' end and AG at the 3' end of introns. At the 3' end of the gene, there is a polyadenylation signal, commonly ATA. Step 2. Transcription produces a pre-messenger RNA, pre-mRNA, which still contains introns. Splicing then removes the introns, leaving only exons connected together. Step 3. The result is mature messenger RNA, mRNA. This includes a 5' cap, the protein coding region that begins with an August start codon, continues through the open reading frame, and ends as stop codon. Following this, there is a 3' untranslated region with a polyadenylation site, leading to the addition of a poly A tail. Step 4. The mature mRNA is then translated into protein.